I'm going to ask an indulgence of you, if I may. I'm going to ask if you wouldn't mind just casting your minds back to set the stage for what I want to talk to you about. Send your minds back to March of 2020 and sort of consider what we were thinking about as we thought about the new epidemic that was sort of taking shape. Things like masking, vaccine development, social distancing, the amount of uncertainty that we had in our lives at that time. I just want to use that as a stepping off point to suggest that the socioeconomic impacts of COVID made it very clear that disease is a very significant global sustainability challenge. However, as you look at this infographic, it's clear that epidemics have a long history. So why were we not better prepared? The research I'll outline today forms part of the Omics initiative. I'll elaborate on that when I come to discuss scaling, our way of scaling, I should say. But one point I want to make now is that the Omics initiative recognizes that today's big sustainability challenges, especially those linked to climate and environmental change, can be much better understood and modeled when we form a bridge between biological, clinical, data sciences, and the social sciences and humanities. Our team focuses on the most consequential disease humanity has faced, malaria. And today, half of the world's population is at threat from the scourge of this disease. For those paying close attention to the last slide, I can just go back very quickly. Just here. You may still be able to pick it out. But malaria has a very long time depth. It's got an antiquity of about 30 million years. Effectively, we left Africa with it. But what's more important is the future impacts of the disease, and diseases like malaria, but other vector-borne diseases in general, which remain highly uncertain, as we've all experienced not so long ago, due to risks posed by climate change and human actions. So how do we respond? How can we sort of work towards this challenge? Our approach is to take a multidisciplinary, longitudinal approach that really sort of centralizes adaptive reflexive modeling, ARM, which seeks an integrated understanding of malaria, with something that we still don't have after over a century of scientific study. And to drive home the kind of connection with sustainability I'm trying to make here, if we take mortality in Africa specifically, 90% of which is actually in children under 5. So there's a constant attrition of a portion of society that become the new leaders, the new educators, the new academics, and so on. So there's really a very complex social problem that we're facing now with many-to-many relationships. The social sciences, for their part, for their role, have shown us that disease is much more complex than the direct impacts on the body. So we've developed ARM as a way to disentangle and study the relationships between malaria, environment, and human actions. And here I just sort of put up deforestation, monocrop agriculture, things like dam building, and of course our ever-expanding scales of urbanization. And in general, please forgive me, this is a generalization, but modern studies do tend to focus on single parameters, such as temperatures. There's very good work on the relationship between temperature and mosquito activity, for example, by colleagues here at Stanford, Erin Mordecai, for example. But these are done over relatively short time depths because it's difficult to gather these data sets. And we believe this is really insufficient to truly understand the scale and complexity of this disease. Just keep in mind, 30 million years. This disease has affected everything about our society, even about our genomes. There's more that we need to do. So we broaden the temporal and spatial context by looking at land use, mobility, policy, transmission rates over hundreds of years. And then we assess how the interrelationships between climate and culture either mitigate or exacerbate epidemics. We gather data from many sites spanning northern and southern hemispheres, including places like Mauritius, which Scott kindly led us to. And I'll focus on one of our sites from that island in the next slide. But we also work in Italy. We have new locations in the Balkans, where there were areas of the environment known to kind of be characterized as plague valleys, and also Zimbabwe, which is a case study of endemic malaria for our project. The time period we study, 300 to 500 years, was characterized by colonialism, which led to massive landscape change, huge demographic flux, and of course, major epidemics. One of which, and I just come back to Mauritius here, one of which hit Mauritius in 1867, wiped out 10% of the population on an island entirely reshaped by colonialism, so 96.4% of the forest was denuded, replaced by monocrop agriculture, and with no indigenous population. So we know exactly when malaria arrived, effectively. And this led to the establishment of this cemetery here, the Bois Marchand Cemetery, which was the largest in the southern hemisphere when it was institutionalized. 400 acres. And the chance to actually work on this cemetery it's just remarkable for us because it's probably one of the best natural experiments in epidemic malaria. And we've got overlays of data from the historical archives as well, which I'll sort of come back to in a second, as well as maps. So our archives actually are from 1868, giving mortality information, locational information, and so on. So there's huge capacity to overlay different lines of evidence over a very distinct time depth. I also want to emphasize the benefits of using data with this type of time depth. Our data sets are extremely large and robust. For example, we have over 2,500 skeletons from which to assess demographic and disease contexts, diet, working conditions, and these can be studied alongside human parasite and vector DNA, as well as archival records on disease, climate, demography, and as I said to you, I'll come back to the burial records in a second. But also sediment cores and coral cores. These reveal critical evidence of climatic and ecological change, as well as landscape responses to agriculture. So we're able to overlay as broad a sweep of lines of evidence that are going to help us tackle this problem as possible. I just want to highlight some of the kind of innovations from the project itself. Our team will be able to overlay historical records of malaria deaths, as I mentioned to you earlier, archival temperature records with evidence of agricultural impacts from things like coral cores. And remarkably, now you might look at this type of data and say, well, it's fragmentary. We've got gaps, we've got problems. Well, remarkably, all of these data sets are accurate to the monthly scale. So this allows us for a unique synthesis of quite disparate data sets, which potentially allows us for the first time to be able to concretely link and correlate mortality with land use, something which is very hard to do today. Two of my colleagues here, this is a sediment core that they've just extracted. So we anticipate recovering the egg casings of the mosquitoes themselves from soil cores like this from our archaeological sites. This provides the DNA of the specific species of vector, but already aligned within a datable climatic profile from the core. And this is really crucial for understanding transmission and as it relates to ecological flux. But for us, perhaps the furthest reaching methodological innovation, is our use of artificial intelligence to analyze our data. We have massive, complex, interdependent data sets. And we've effectively tried to advance the types of tools that you're familiar with, that Google uses, effectively studying your search history to train its algorithms, where we literally use history, as well as climatic, archaeological, genomic data to train our algorithms to seek patterns between outbreaks, mortality, vector, climate, and human activity. Our AI is designed to be predictive, and we develop policy-relevant algorithms. And this work can be, sorry for this kind of complex site, but I just want to point to historical records. This work can be incredibly revealing. Just working on the historical archives, we had a young data scientist, 16 years old, working with our team, just did some phenomenal work, and basically showed that if you were going to a hospital at that time with malaria, you were more likely to die than if you'd stayed at home. This was completely counterintuitive. And as it turned out, when we tried to understand the social side of the social underpinnings for this, as well as the scientific, the social was relatively straightforward. Most of these people dying from malaria at this time were indentured laborers. So they were under contract. If they missed a day's pay, they were docked a day's pay, and they got into a cycle of debt. So you didn't go to hospital unless you were desperately in need of it. There's a scientific component, though. Malaria, a research paper I read, it seemed to suggest that malaria sort of goes into a state of depression. It's depressed somewhat in the body under conditions of malnutrition and, of course, dehydration. So you can see why that happens. The body's not circulating the blood in the way that it should. when you look at those 2 points together, without this kind of complexity, without using these AI tools, we simply wouldn't be able to see this type of outcome from the archaeohistoric record. And it seems like it's something, an archaeological conundrum. Right here in the USA, during periods of COVID epidemics, we were seeing the same thing. Poorer communities were not wanting to go to hospital because of associated costs. And so this is a really important aspect from which we can actually start to understand the economics of pandemic and how we can form a bridge between past and present. There's also a fairly massive untapped repository. Masking, social distancing, responses to vaccines, as well as misinformation, ostracism, none of these things are new. So we have a huge trove of evidence on social norms before, during, and after epidemic disease. And this is really critical for us to prepare ourselves for the next pandemic, which is estimated is likely to be more consistently reoccurring because of climate change and so on. I just want to take a pause here. That's all I'll say about malaria for a second. Very quickly want to mention that we have a Rift Valley Fever project working with colleagues in medicine on basically connecting pastoralism, people and policy, and how colonialism has left legacies on human environmental interactions that cause major harm to populations today under extreme weather conditions. But also we're undertaking a study, again using the sediment core, but looking at the entire pathogen DNA load across about 2,000 years with coastal communities in Madagascar. And I'm going to have to rush the last bit and just say, where are we taking this approach? And I want to focus on the approach here. How are we trying to scale? We have many academic partners, both here and overseas. We also work with government and parastatal bodies. We work with local NGOs. And all of our work is really deeply embedded in local community. All our permitting and so on is done with local community, of course. But we've taken our conversations further. We're working with, we're speaking to colleagues at Gates, sort of understand how we might take our work to policy, but also how we might integrate these huge data sets that we have into contemporary quantitative analysis and modeling. We've also spoken to Stephen Hoffman at Sanaria. I just want to bring you back to that, genomic change over climatic and aligned with climatic conditions. Well, that was really exciting to them because this would be very helpful to developing the next generation of vaccines, which are cognizant of climatic influence on the genomes of mosquitoes. And just very quickly, I mentioned to you, I'd come back to the Omics Initiative. This approach to research has been seeded into the Omics Initiative, which is effectively an incubator. And we want to basically generate an institutional home, a center for research that is applied, directed towards policy interventions, while emphasizing the power of using multiple sources of data to provide a longitudinal lens on the modern condition. Why omics? Because we integrated genomics, phenomics, and proteomics. We've been well supported at Stanford, particularly by IRIS. We've had a soft launch in 2022 of the Omics Initiative. We've also had external support, and we're currently preparing two major grants, an ERC grant, which is a 10 million euro grant with 4 institutions, and we're Stanford's nominations for this predictive intelligence for pandemic prevention, which hopefully we've been well supported by colleagues at VPDOR. Rob is one of the co-PIs. It's very aspirational at the moment, but we want to concretize and scale to a center initially focused on longitudinal studies of disease specifically, but hopefully having a home in SDSS. I just want to summarize, sorry, I'm running short of time, with the last 2 slides, summarize and conclude by coming back to malaria. ARM represents a paradigm shift in the way we study how disease impacts society. It's applicable to other vector-borne diseases, as I've shown you with the case study in Rift Valley Fever. However, the concept seeded into omics could help us tackle a class of problems by being applied to other human environmental challenges, principally those around climate change and land use. I'd like to ask you, how could, thinking about this from your own research, thank you very much, how could a data-driven understanding of the past help my work? How could our approach be useful in your fields? And ultimately, the greatest sort of transformative impact that we see is that the Omics Initiative illustrates how we can effectively operationalize the past for the future benefit of humanity. And with my last slide, I hope that's clear, I want to bring us back to a point, thank you very much, raised by Dean Arun at the first Coordination Committee meeting this year. Where are our blind spots? I believe we need disciplines like archaeology to help offer context to core questions like, where do our sustainability challenges stem? The discipline also has to step up. This is not a typical archaeological project, but we really need to use these disciplines to help us kind of contextualize our work more effectively. This work is also important because because of the thematics involved. Decolonization is at the core of this work. Social and environmental justice aligned are at the core of this work. The human dimensions of sustainability at the core of this work. And effectively, we risk failure if we don't recognize that data detached from human behavior will invariably lead to solutions that are not aligned with society's needs. It may look like there's a meeting point in the future, but that's just an illusion. Thank you.